Hi there, are you wondering what vitamin is? Do you know how many types of vitamins there are? Actually, there are various vitamins in our lives. In this video, we will tell you what a vitamin is, how to classify vitamins, and what is the source of vitamins. So first, what is a vitamin? Vitamins, by definition, is an essential micronutrient that comes from natural food, required in minute amounts, cannot be synthesized by humans, essential for growth, development, and maintenance of the human body. Then, how to classify vitamins. Vitamins are classified based on whether they dissolve in fat or water. Now, let's begin with a fat-soluble vitamin. Fat-soluble vitamins are absorbed along with dietary fat, and once inside the body, they are transported with fat. Fat-soluble vitamins, however, are not excreted in the urine, but are stored in the liver and fatty tissues. There are 4 types of fat-soluble vitamins, namely vitamin A, D, E, and K. Vitamin A. Did you notice, for generations mothers have been telling their children that eating carrots will help them see better in the dark? It turns out that they were right. Carrots are a good source of beta-carotene, which can be turned into vitamin A in our body. Vitamin A is also important for the immune system, because it is needed for the differentiation of cells into the various types of immune cells. Vitamin A can be found in the diet both as retinoids and carotenoids. There are 3 different retinoids in the body: retinol, retinol, and retinoic acid. Retinol is the immediate precursor to 2 important active metabolites: retinol, which plays a critical role in vision, and retinoic acid, which serves as an intracellular messenger that affects transcription of a number of genes. Vitamin A in the diet comes from both animal and plant sources, such as liver, fish, egg yolks, dairy products, carrots, squash, and apricots. However, vitamin A can be resistant at 100 degrees Celsius, oxidized fast at 150 degrees Celsius and above, and also oxidized easily by UV light, air, or oxygen. Vitamin D. Vitamin D is needed for calcium absorption, and to maintain the proper ratio of calcium and phosphorus in the blood. Vitamin D is not widespread in the food supply. Natural sources include egg yolks, liver, and fatty fish, such as salmon. Foods that are fortified with vitamin D include milk, margarine, and some yogurts, cheeses, and breakfast cereals. In addition, vitamin D can be obtained through our skin when we are exposed to sunlight. This is why vitamin D is also known as sunshine vitamin. Vitamin D can be categorized as a stable vitamin, since little or no loss of vitamin D during processing or storage. Vitamin E. The chemical name for vitamin E is tocopherol. There are several forms of vitamin E that occur naturally in food, but only alpha-tocopherol can meet vitamin E requirements in humans. Vitamin E functions as an antioxidant that neutralizes reactive oxygen compounds before they damage unsaturated fatty acids in cell membranes. Vitamin E is also important in maintaining the membrane integrity of red blood cells, cells in nervous tissue, and cells of the immune system. Dietary sources of vitamin E include nuts and peanuts, plant oils such as soybean, corn, and sunflower oils, leafy green vegetables, wheat germ, and fortified breakfast cereals. Vitamin K. Vitamin K is essential for blood clotting to prevent excessive blood loss when skin is broken. Vitamin K is also needed for the synthesis of several proteins involved in bone formation and breakdown. If you do not know, bacteria that live in our intestines produce some of the vitamin K we need, but some of it must also come from the diet. The best sources are leafy green vegetables such as spinach, broccoli, Brussels sprouts, kale, and turnip greens. Vegetable oils including soybean, cottonseed, canola, and olive oil also provide vitamin K. Others such as meats, milk, and eggs provide little of this vitamin. However, for the stability, vitamin K is significantly affected by heat and sensitive to both fluorescent light and sunlight. Let's move to water-soluble vitamin. Water-soluble vitamins dissolve in water. Body stores only limited amounts of them, so they need to be taken daily, and they are readily eliminated in urine. Let's begin with the vitamin B complex. Generally, vitamin B complexes are required for essential metabolic activities as part of active enzymes, and the sources of vitamin B usually can be found in the same food sources such as liver, yeast, and bran of cereal grains. Vitamin B1, thiamine. Thiamine, like the other B vitamins, is a coenzyme. The active coenzyme form of thiamine is needed in the body for the production of ATP from glucose and for the synthesis of acetylcholine, which is needed to transmit nerve signals. Thiamine is also needed for the metabolism of other sugars and certain amino acids, and for the synthesis of ribose, a sugar that is part of the structure of RNA. For the stability, it is stable to oxidation and light, but least stable in solution at neutral and alkaline pH. Vitamin B2 or riboflavin. Riboflavin is needed to produce ATP from carbohydrates, fat, and protein. The active coenzyme forms of riboflavin function as electron carriers in the citric acid cycle and the electron transport chain. Riboflavin is also needed to convert a number of other vitamins, including folate, niacin, vitamin B6, and vitamin K, into their active forms. For stability, riboflavin is sensitive to light. If the milk is in a container that exposes it to light, much of its riboflavin will be destroyed in just within 2 hours. Vitamin B3, niacin. Niacin coenzymes are electron carriers that are needed for energy metabolism and in reactions that synthesize fatty acids and cholesterol. There are two forms of niacin, nicotinic acid and nicotinamide. Both of them can be used to make active niacin coenzymes. Niacin can be synthesized in the body from the essential amino acid tryptophan, but the synthesis is extremely slow and requires vitamin B6. Nicotinic acid and the corresponding amide are probably the most stable of vitamins. Vitamin B5, pantothenic acid. Pantothenic acid is part of the structure of coenzyme A. Vitamin B6. The chemical term for vitamin B6 is pyridoxine. Vitamin B6 is needed in its active coenzyme form for the activity of more than 100 enzymes involved in the metabolism of carbohydrates, fat, and protein. In addition to its role in amino acid metabolism, vitamin B6 is important for the immune system and for the synthesis of hemoglobin, certain neurotransmitters, and lipids that are part of the myelin coating on nerves. Vitamin B7, biotin. Biotin is a B vitamin that plays an important role in the citric acid cycle and is needed for the synthesis of glucose and the metabolism of fatty acids and amino acids. Biotin appears to be quite stable. Heat treatment results in relatively small losses. Biotin is also stable to air and at neutral and acidic condition. Vitamin B9, folate. The B vitamin folate is needed for the synthesis of DNA and the metabolism of some amino acids. Folic acid is unstable to heat, air, water, and alkali, and careful cooking methods must be employed to ensure adequate amounts of this vitamin are retained. Vitamin B12, cobalamin. The most complex structure of the vitamins. Vitamin B12 is a B vitamin that is found only in animal product. Vitamin B12 is not destroyed to a great extent by cooking. Okay, now, we will further study about vitamin C. We choose to further study in vitamin C because vitamin C is one of the well-known vitamins, which plays an important role in facilitating body function. Vitamin C, consisting of ascorbic acid and dehydroascorbic acid. This ascorbic acid is highly polar and readily soluble in aqueous solution. The best-known source of vitamin C is citrus fruits, such as oranges, lemons, and limes. Other good sources include strawberries, cantaloupe, leafy green vegetables, and vegetables in the cabbage family. There are several biological functions of vitamin C. First, this vitamin C is needed for the synthesis and maintenance of collagen. Collagen is the most abundant protein in the body and the predominant protein in connective tissue. Without sufficient vitamin C, collagen cannot be synthesized or maintained. This causes the symptoms of scurvy, which include poor wound healing, weakened blood vessels, bone fractures, bleeding gums, and loose teeth. Vitamin C is also needed for the synthesis of neurotransmitters, which convert tryptophan and tyrosine to serotonin and epinephrine. Besides, vitamin C also enhances absorption of iron. Ascorbic acid reduces the inorganic ferric to ferrous form, which is easier for our body to absorb. Ordinary absorption of iron is quite poor and may lead to iron deficiency anemia. Vitamin C also acts as highly effective antioxidant. It protects proteins, lipids, carbohydrates, and nucleic acid from damage by free radicals. Vitamin C also synthesis of carnitine, which is essential for the transport of fat to mitochondria for conversion to energy. However, vitamin C also has its own stability. The predominant pathway of vitamin C degradation in aqueous liquid systems entails oxidation of ascorbic acid to dehydroascorbic acid, which itself promptly degrades to 2,3-diketagulonic acid. By the hydrolysis of dehydroascorbic acid, the molecule loses its vitamin property. This oxidation of ascorbic acid to dehydroascorbic acid depends on First, effective pH. Vitamin C content stable at low pH and significantly reduced in high pH. Second, effective temperature. Vitamin C content loss with the increased heating time and heating temperature. Third, effective heavy metal ion. Some heavy metal ions will reduce the stability. Some will increase stability of vitamin C. For example, copper will decrease the stability of ascorbic acid as copper catalyzes the redox reaction and increases the rate of ascorbic acid convert into dehydroascorbic acid. Fourth, effective oxygen. Ascorbic acid that is exposed to oxygen will oxidize faster. This is because oxygen is a good oxidizer and ascorbic acid will leach out from the cut or bruised surface as well. Application of vitamin C in food processing. First, inhibits non-enzymatic and enzymatic browning. Antioxidants such as ascorbic acid will prevent the formation of brown pigment by reacting with intermediate and reduced quinones back to phenols. Second, it plays a role in the dough system, which improves loaf volume and bread texture by reduction of dehydroascorbic acid back to ascorbic acid. This drives the reaction of sulfhydryl compounds in the gluten to form intermolecular disulfide bonds, and able to retain carbon dioxide. The recommended daily intake of vitamin C, depending on age, gender, size, and life stage as well as health status. For example, 65 mg per day of vitamin C needed for adolescents, 70 mg per day for adults, and 85 mg per day for pregnant women. But a tolerable upper intake level for vitamin C is 2000 mg per day. Furthermore, do you know the myths and the truth about vitamin C? First myths, vitamin C can be only found in oranges. The truth is citrus fruits such as oranges are an excellent source for vitamin C, but other foods such as vegetables also provide us with vitamin C as well. Second myths, vitamin C provides us with energy. The truth is vitamin C does not provide us energy, it assists our body function. Third myths, vitamin C can prevent and treat common colds. But scientific studies have shown that vitamin C does not reduce the incidence of colds, but can reduce the duration and severity of cold symptoms. For the analysis of vitamin C, there are several types of vitamin C analysis. First, spectrophotometry that involve UV light. Second, redox titration that involve redox reaction. For example, iodine titration method and indophenol titration methods. Third, chromatography method that involve both mobile and stationary phase, which separate the mixture of molecules present. Fourth, electrophoresis that utilizes capillary tubing, high electric field strengths, and detector. And fifth, enzymatic methods for enzymatic reaction. However, in this video, we choose the high-performance liquid chromatography method. This is because it is more accurate, sensitive, able to specify compounds that want to analyze, and in a shorter time. First, sample preparation. The homogeneous solid sample of orange was measured around 10 gram to 30 gram, and mixed it with 60 to 80 milliliter 3% of metaphosphoric acid for 1 minute. The obtained extract was filtered through filtration paper, and washed it for a few times by using vacuum pump filtration. Next, the filtrate quantitatively transferred into a 100 milliliters volumetric flask and 3% of metaphosphoric acid was added up to 100 milliliters volumetric mark. All the sample solutions were filtered again through a 0.45 micrometer syringe filter. After that, the samples were run in the HPLC system. For the analysis, the reversed phase liquid chromatographic method was used for determination of vitamin C. Consisted of an isocratic elution procedure with photodiode assay detection at 254 nanometer. The mobile phase used was a mixture of methanol water. The flow rate of the mobile phase was 1.0 milliliter min 1 and 20 microliter injection volume of samples and standard were used in quantitative analysis. The temperature of the analytical column was kept constant at laboratory temperature, which was 25 degrees Celsius. The standard of the vitamin C was an external standard, which 1 milligram per milliliter natural vitamin C derivatives. For the identification, it was performed by the comparison of retention time of analyte in the analyzed sample with the retention time of the calibration standard. Quantification was carried out with the external standard method using the following equation formula to calculate the vitamin C concentration on samples. And lastly, this is the references we used. Our task distribution.
Hello everyone, my name is Ong Keen with matrix number 204002. In this video, I have involved in finding the content, especially for the introduction part. And then I search for useful pictures and videos to include in our video and do a rough design for our editor to do the final editing Okay, that's all. Thank you.
Hi, my name is Darren Sim Xuan Yu with metric number 201777. In this video, I find the information about vitamin C and also analysis part of vitamin C, and I help some in designing this video as well.
Hamzah, metric number is 201901, and my contribution in this video is that I could say almost all the related I already. I find the related pictures and videos to our topic, but of course Ong and Darren also help me by giving their opinions and also help me finding the related pictures and videos to our topic. I also have been finding the content of the videos, but mostly done by Ong and Darren. Yeah, we is very meeting to make sure our flow of these videos and flow of the content is going well. And yeah, I would say everyone contribute our parts and we help each other to make sure this video is well presented.